Welcome everyone to this IMAG working group presentation by the Systems Biology Working Group. I'm pleased to welcome Dr. Simon Kahan and to this webinar here, and he and his colleague Seunghwa will
All right.
Biocellion, accelerating computer simulation of multicellular biological systems models. Simon, please go ahead.
Okay, I want to make sure that Seunghwa is able to display the slides, but just as a very quick introduction, actually, why don't you introduce yourself while I set up this, get this thing working?
Okay, so I'll talk about myself first, like before Simon fixed some technical glitches. So I'm basically, my background is in high performance parallel computing, but like now I joined after finishing my PhD, I joined the computational biology group at Pacific Northwest National Laboratory, and like during my postdoc period, I was sitting at Institute for Systems Biology. So I worked in a collaborative project between Pacific Northwest National Lab and Institute for Systems Biology. Then, after becoming a staff scientist, I moved back to Richland and started to work with a computational biology group at PNNL. Then now I come back to Seattle, and I'm sitting at Northwest Institute for Advanced Computing. It's a joint center between PNNL and University of Washington, and now I'm working here at the intersection of high-performance computing and computational biology, and working on developing the Biocellion framework, something I'm going to talk about today. And now presentation is ready, so like Simon.
I'll be very quick. So I'm right now affiliate professor at the University of Washington Computer Science Department, and also affiliated with the Institute for Systems Biology. Before that, I was an employee at the Pacific Northwest National Laboratory, where I started the Biocellion project a few years ago with Ilya Shmulevich at the Institute for Systems Biology. And we brought on Seunghwa to do the coding. So he's the most appropriate one to give this talk. My background is high performance computing. I got my doctorate in computer science from the University of Washington in '91 and spent most of my career at Terra Computer and Cray, and then Google for a couple of years before joining the National Laboratory. So, in the interest of time, we just get started.
Yeah, but sorry for all the delay. And like, if the resolution of presentation slides isn't high enough, you can download the original slide in zip file from biocellion.com. So you can try that. And I'll move to the presentation. So I'll talk about Biocellion. This is a software framework to accelerate large-scale discrete agent-based simulation. But before jumping to the Biocellion framework, I'll give my high-level thoughts about biological system modeling, which heavily affect the design of Biocellion. Then I'm going to present high-level design principles of Biocellion, move to some details, present some early research, and I'm going to talk about how this framework will going to evolve over time. So like biological system modeling, like I may not need to explain this to this community, but I'm going to say that anyway. So basically, the main reason we are doing this is to really understand, predict, and manipulate behaviors of important biological systems, something like predicting cancer prognosis or filtering out drug candidates, like finding the best yeast strains for biofuel production, or like really predicting what is going to happen, like if we put some like genetically engineered bacteria to some site for environmental remediation. But like these are like needless to say something very, 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 very difficult problems, and something we should work on for coming decades to solve. So like, how can you actually get there, solve this very, very, very difficult problem? So like, the way we should go is first start with some simple model, then keep incrementally updating that model to make it more and more accurate. And to really get there, what is important is we should be able to directly compare the result of simulation to well-lab experiments. So like we can validate every incremental update as long as we are moving to our final goal. And there are three principal approaches in modeling large biological systems. Like one is population-based modeling, so like this focuses more on the average behavior of cells in certain cubic boxes, and discrete agent-based modeling that maps a cell to a discrete agent or more than one discrete agents, and one can mix these two approaches. But like to really support the incremental updates, my belief is like we should use same approach for both simple model and something highly complicated model. And like if we adopt population-based modeling, it may work for simple cases like we just describe cell behavior based on their sort of average characteristics. But like once we want to add more of cell-to-cell variation and more complex knowledge about how a single cell behaves, like this approach becomes completely untractable. For example, something like if we want to add a biological rule saying, like if a stem cell divides, like something like 80% of case, it goes through a symmetric division, like producing one stem cell and one non-stem cell, and like 20% of cases, it actually goes like symmetric division, and so it ends up with two stem cells. Like this kind of biological rule is very easy to add with discrete agent-based modeling. We just need to generate one random number, and based on that number, we can go to either symmetric or asymmetric division. But like with population-based modeling, it is much more tricky and non-trivial how to incorporate this knowledge into the model. And like if we try to add more and more of this kind of cell-level rules, like population-based modeling becomes totally intractable. So in that regard, like discrete agent-based modeling has some benefit. And also we want to simulate in the scale that matches like wet lab experiments. So like most 2D models, except for like if 2D model really well represents what's happening in the real world, which is mostly not a case because in real world everything is 3D. So except for so most cases, we want to simulate things in 3D and also matching the scale of well-lab experiments. So this is something desirable, but like
check to make sure. Everybody lost her. Have you guys lost the slides?
Jeff, please stop sharing your screen. So if you guys might have to click and
reclaim it.
Yeah.
Okay.
Okay, but the problem is this is computationally expensive. So like to really match the scale of real biological systems, we need to simulate millions, billions, or even some cases trillions or more of cells. And many people are saying like simulating this number of discrete agents is computationally intractable under practical limitations. So this is the computational complexity is really barring us from moving in this direction. Okay, so computationally intractable under practical limitations, but that's really true. And my background is in high-performance computing, and the HPC community has totally different criteria for what is computationally tractable and what is not. So basically, one of the current largest supercomputer is Titan from Oak Ridge, and like that machine has something like millions of cores. So like if we want to simulate millions of cells, it's roughly saying just few cells per core. And like if we want to simulate something like billions of cells, it's just something like a few thousand cells per core. So in that, and this community and the HPC community is also trying to build a much larger system, and like computing cost typically goes down with time. So like they are trying to build something like billion core machine and within coming 10 years. So like billion core machines, like some billion cells is just few cells per core. So like if we can efficiently use this kind of machine, what has been considered as computationally intractable can actually become computationally tractable. But the key problem in efficiently using this kind of machine is software. So writing software for that kind of machine is very, very difficult. So like what HPC people do to efficiently use that kind of machine is first selecting efficient and scalable algorithm. And those algorithms run fast, but typically much more complicated to implement than the brute force approach. And then people typically tune such code for the machine. And like this exhaustive performance tuning can easily add 1 order of magnitude speed up. Like this is a research from the work I did in my first PhD year, and like with all the performance tuning, like I can get 34 times or 50 times speed up, and this isn't something that exceptional in the HPC world, something that happens very common, but this clearly is very, very labor intensive and requires a lot of time investment. And making this all work well on computer with millions or something billions of cores, which will come in the future, is very, very, very difficult and very, very time consuming. So like HPC can be extremely powerful, but it's also very difficult to use. So like to really properly use this kind of system, you really need a group of hero programmers with PhD in high performance computing or something equivalent. So this isn't something most computational biologists can easily use. So now I'm going to move to the Biocillion framework. So the goal of this project is making those extremely powerful, but also very difficult to use HPC computers usable for computational biologists without really requiring HPC expertise or investing a huge amount of time to implement software. And there are always trade-offs. So basically, flexibility means like flexibility to express arbitrary mathematics, and so like system gets more and more so like if the framework gets more and more flexible, users can bring their own arbitrary mathematical rules to define biological system behavior. Well, like then users need to do more to properly set up a model so that somewhat lowers usability. And also, if I ask user to provide more, then negatively affects performance. So like basically, one of the biggest challenge in designing Biocillion is managing this the trade-off between flexibility, ease of use, and performance. And making this trade-off, my top priority is supporting serious computational biologists who want to improve the state of the art. So like to really get there, we really need to allow them to express their own mathematics rather than adopting just some off-the-shelf ready-made rules. So like the top priority is in flexibility, but I'll try to make this still usable without spend a huge amount of time in programming or going like spending multiple years to learn high performance computing. And performance is clearly the main selling point of this framework. So I try to get maximum performance within that limitation.
Just so you know, it's almost 10:30, so no one has to speed up.
Okay. But like providing that flexibility is not easy because biological systems are very diverse, and like even for a same biological system, models are also diverse, and also our understanding of biological system changes very fast, and models evolve fast accordingly. So if we develop software just assuming a single model, it's not going to last for very long. And like for me, like developing this Biocillion framework took 3 years. Like I really devoted my past 3 years to develop this framework. But like typically models lifespan is much shorter than 3 years. So it'll be not just dead on arrival, but like, if I design this framework for a single model, it'll be dead even before it's finished. And like developing HPC software is very expensive, and that cost cannot be justified. So Biocillion approaches, okay, in many models, there are some parts that keep changing or different from every model, but there are something common. So like for that changing part, Biocillion asks users to provide rules, something like, okay, what are going to be the behavior of individual cell, or if two cells interact by touching each other, what are going to be the outcome of that interaction, or like tracking molecular concentrations in the extracellular space by solving PDEs, how one should set PDE parameters, like ask users to provide this kind of rules, but like the framework will gonna manage parallel computing related changes, like something like managing parallelism and providing efficient solvers that is fast, but some can be pretty complicated to implement. So basically, this framework considered interactions between cells, interactions between cell and the environment, and also individual cell behaviors. So I'll move to some details. So basically, discrete agents are key players in Biocillion simulation, and also there's the environment. So discrete agents, like model developers can assign an arbitrary number of state variables to every discrete agent type. So by default, as a sphere, but like one can add direction vector and height, then one can treat that discrete agent as a cylinder. And also can add another variable to add more internal states. And also for the environment, like for each grid box, users can add an arbitrary number of variables. And for some region, like we need to simulate all like cell behaviors, cell-cell interactions, and we also need to solve PDEs. But like for some region, that region might be only relevant in solving PDEs. Something like if we are simulating yeast cells growing on top of very thick agar, then that agar part is mostly relevant in just tracking molecular concentrations, and unless cells invade into agarose gel. So for that kind of region, like Biocillion allows to set those regions as PDE buffer, then only data structures relevant in solving PDEs are maintained for that region to save memory and some computing. So here's some example code of setting up and providing information about each discrete agent. And like here, like users can assign an arbitrary number of like real type variables or int type variables. So I'm not going to go through every detail here. And evaluating direct cell-cell interactions. So like cell-cell interaction, there can be two different interactions, something direct, really by touching other cells, and indirect through like molecules, and are considered direct interaction first. So like to evaluate direct interactions, one thing we need to do is enumerate every pairs with an interaction distance. So for each cell type, we asked users to provide maximum interaction range, and for two different cell types, we consider the average of the two interaction range as the interaction range for two cells of two different types. And Biocillion's job is enumerating every pairs within this interaction range. But like once those pairs are given, and Biocillion also gives the environment information each cell belongs to. But based on that information, like evaluating pairwise interaction is user's job. So like if you move to code, like users are given information about two interacting cells and the variables associated the two boxes the two cells belong to. But based on that, like updating force variable or some additional extra variable, something relevant, that'll be user's job. So like in case something like if some cell is like we want to model cells getting compressed. So like in that case, the net force might be small, but like if we sum the absolute value of force, then that force might be big. Then we can sort of figure out that that cell is compressed, and we may set that cell will gonna die because it's under too high pressure. To model that, like users can add additional variable to sum the absolute value of force, and to do that, Biocillion allows users to add that variable to model, that additional variable to be updated through evaluating mechanical interactions. And also, Biocillion supports indirect cell-cell interaction and cell environment interactions. So basically, users can update variables associated to the grid boxes by model-specific rules or by solving PDEs. So if they want to update them based on model-specific rules, they can access all the variables associated with a box and its 26 neighboring boxes. And based on that, users can bring their own rules to update the variables associated with the box and its neighboring boxes. And for solving PDEs, like Biocillion supports adaptive mesh refinement, implicit method, and implicit method based on multi-grid. So basically, user's job is tagging the desired each box with the desired adaptive mesh refinement level, and users are asked to set PDE parameters. But based on that information, generating the AMR adaptive mesh refinement hierarchy and solving the PDEs efficiently is Biocillion's job. And like, if I exaggerate a bit, like the HPC community is all about solving partial differential equations, and there are several efficient existing solvers, and like Chombo from Lawrence Berkeley is one of that. So I adopted this and heavily customized that to work well in the Biocillion's context. So the back end of Biocillion's PDE solver is Chombo from Lawrence Berkeley. And so like this is an example code to update like variables associated with grid boxes based on model-specific rules, or users are asked to set diffusion coefficient, or in this code, users tag each box with the desired adaptive mesh refinement level. So these are the information users should give to the framework. But like based on this, actually solving PDEs is the responsibility of the Biocillion framework. And also users are asked to provide rules for it to determine individual cell behavior. And currently, like each cell behavior is evaluated sequentially. So parallelism comes from the many number of cells. And writing that code is mostly up to users. But like, what if users want to model cell regulator network using deterministic ordinary differential equations? Biocillion provides solvers, and we adopt ordinary differential equation solver from Intel for this purpose. So here's some code examples, like users can update the state of state variables associated with cell based on their own rules and simulating biological systems, there are multiple processes, and different processes require different time step sizes. For example, like evaluating physical interactions between cells and tracking cell movement, we may able to use a bit larger time step size, but really coupling the changes in the extracellular space and changes in cell state, those may need to be coupled more tightly. And solving partial differential equations often requires smaller time step size. So basically, Biocillion maintains 3 different time step size for different purposes. And we are assuming that like the direct users of Biocillion are serious computational biologists with strong mathematical modeling background. So they may have ability to decide time step size maybe better than us. So like we ask users to determine the time step sizes, but actually managing parallelism and data movement based on those different time step sizes is a responsibility of the Biocillion framework. So like I'm gonna start with the sort of the hello world example of multicellular biological system modeling. So that is cell sorting. So assume like we have 2 different types of cells, like red cells and green cells, and each cell is mapped to a sphere. And like if there's overlap between 2 spheres, they shove each other. And like if they are a little distant and they are of same type, then there's adhesion. But like there's no adhesion between 2 cells of different types. So like this is the core code modeling this. So basically, if there's overlap, then there's shoving and there's adhesion between 2 different cell types. Yeah, seems like video isn't working optimally.
Oh, it's working fine, isn't it?
I know, but like it's cube, it's not.
Oh, it's distorted.
Yeah, it's distorted. But like if you downloaded the zip file from the homepage, you can see the proper video. So like here, like it's a little difficult to figure out sorting pattern, but like if we zoom in, what's happening is something in the right side, and like 60 million cells, that sounds like large, but like this isn't something really that expensive. Like this can be run in a like multi-core workstation, and like we can also easily sort something like 1 billion cells. So for 1 billion cells, like it's very difficult to find any sorting pattern because like this sort of typical patch will be just a dot and 1 billion cells. And 1 billion cells, we can do that in something like small cluster with something like 16 nodes. So, and are going to move to something weird, and there are multiple early adopters who developed their models using Biocillion. So this is a work from Winning Show's group at Fred Hutchinson. So they considered 2 different strains of yeast cells growing collaboratively. So like red cell produces adenine and consumes lysine, and green cell produces lysine and consumes adenine. So like, and their collaborative relationship promotes intermixing between two different cell types, and like these cells grow on top of very thick agar compared to the size of the cell. So their original model maps a cell to a fixed size cubic box. So like this mapping map might be okay for their problem, but like say like if we want to incrementally update the model, like and add rules like if a cell divides, like mother cell is bigger than the daughter cell, or like if we want to add certain rule about their dividing direction. Like the original model does not allow adding these rules in a straightforward fashion. So this really doesn't allow incremental updates of the model. But like Biocillion, cells are mapped to a arbitrary size sphere. So adding rules about dividing into cells of two different sizes, or adding rules about setting division direction, they can be integrated in a straightforward fashion. And also modeling diffusion, like basically there are two different modes of cell growth. If two cells are distant, then they communicate and share nutrients through agarose gel. But like once they form a community, then nutrients are mostly shared within that community. So like in the first case, if diffusion coefficient between the cell and gel agar is large, that promotes growth because like more nutrients will diffuse back to the cells instead of going deep down the downside of the agar. But like once they form a colony, then like small diffusion coefficient between the colony and agar promotes growth because less will be lost to the agarose gel, right? But like if grid size is too large, we really cannot properly capture this. For example, something like if grid size is 5 micrometer, like this properly models nutrient sharing via agarose gel. But like if grid spacing is 20 micrometer, then even though they should share nutrients through agarose gel, in this mathematical model, this will more work like the colony growth mode, like sharing the one in the right top side. So like there's a need to adopt like small grid spacing if something like this happens frequently, but like reducing grid size like by 10 times is and 1,000 times more computing in 3D. And the original model adopts the explicit method, and that explicit method has some stability requirement. So like if we reduce time step sizes, reduce grid spacing, we also need to reduce time step sizes. So that ends up like 10 to the 5th increase in computing, and which can be very, very expensive. But Biocillion adopts implicit method, and it doesn't have that stability requirement. And with adaptive mesh refinement and high performance computing, Biocillion can actually make this grid spacing computationally feasible. And also, if something like, so if two cells going to the same box without really touching each other, like if something like this happens only very infrequently, if something like initial cell density is low, this will not going to happen very frequently, then one may be able to use a larger grid spacing to save time, but that can has different artifacts. Like, for example, like here, like if a box is only partially filled, then like only a small fraction of that box can hold nutrients. because most of the box are just air. But like using the original model, it's really hard to incorporate that into this model. And also if boxes are only partially filled, like we need to scale down diffusion coefficient, and Biocillion allows this kind of modification in a straightforward way. So Biocillion allows users to set the box capacity factor, kappa, and Biocillion allows users to scale set diffusion coefficients based on their arbitrary rules.
That's 12 to 11. Just so you know.
Okay.
Yeah.
And here is a video outcome of the simulation, and downloaded the video from the website. Then you can see this growing to a pretty large community.
Can we get anything to work on this at this point? There we go. So it looks like it doesn't have the.
Let's move to the next slide.
All right.
Yeah. You can download videos from the homepage, and you can see that by yourself.
Yeah.
Sorry. Okay, and the second model. Uh-oh, I moved too far. As a bacterial system in soil structure. So like basically there are 3 cell types. So like in the soil there's a polycarbon which needs to be broken down to smaller molecules to be consumed by cells. And there are 3 different types of cells. Like one cell type releases enzyme that breaks that polycarbon to smaller molecules, and the second type express hydrolysis enzyme on the cell surface, and the third cell type is a free rider. And this paper models cell regulatory network controlling these enzyme production and cell growth using deterministic ODEs. So here the equations in the left side are the heart of basically it describes how a single cell behaves. And so Biocillion supports solving ordinary differential equations. So basically users need to translate these rules to this code, but after that, actually solving those deterministic ODEs is the responsibility of Biocillion. And like cells grow in this kind of complex soil structure, and our skip playing video, but like you can download it from the homepage. And the original model, they simulated something happening only on a small fraction of this larger soil structure, but Biocillion allows them to scale this up and move to a larger spatial scale. And I hope I can play this.
No, if you can't, you can always go to the files, right?
Okay,
and I can do that for you if you want.
Okay, but like
all
the videos are available in the zip file. So
it's gone.
Yep. So this is a work from Ryan Tassef at Institute for Systems Biology, and he's working with some French institute to model microfluidic device, and like he has outcome from experimental research, and he built a model in the same size. And can you play it?
Just click it. There you go.
Yep, and like this is a simulation outcome, but like what is important here is he adopted very complex mathematics to properly model cell budding directions, and like that involves torque and something pretty complicated. So one really cannot integrate that kind of mathematics unless like the framework has this level of flexibility and allows users to express their own mathematics through coding. So Biocillion somewhat sacrificed usability by forcing users to do code, but like that also allows this kind of modeling. And he also built some prototype skin model, and he considers cells of different shapes. So I didn't know very much about this model, so I just moved to the next. Wrinkle formation video, and in the second video you can see smiley face, something that is visually pleasing. And like if you download the zip file, you can try that on your own. And this is a prototype model for ductal carcinoma growth or microtumor growth. And Bill at PNNL is building maybe the most complicated single-cell model we have, and he's trying to model single-cell behavior based on thermodynamics rules instead of using some kinetic modeling, some kinetic reactions. And like this model, even simulating a single cell can be pretty expensive, but using Biocellion and using large parallel computers, he can scale this to a multi-cell simulation. So that is the current status of Biocillion. But like but there are more to be added to really support incremental updates matching the scale of real biological systems. For example, in simulating soil structure, if structure gets larger and larger, we really cannot ignore like water flow. And also for microfluidic devices, like for large microfluidic devices, like advection is very important in long-distance communication. So to really support that, we need to add solvers for Navier-Stokes equation to figure out the speed of velocity of the flow, and also advection reaction diffusion equations to update molecular concentrations based on that velocity. And fortunately, like Seunghwa already have solvers for Navier-Stokes and advection reaction diffusion equations. So like to integrate this to Biocillion, we need to customize those as I did for reaction diffusion equations. And we are also interested in supporting blood flow modeling in microvasculature, as it affects delivery rate of nutrients, oxygen, drug molecules, or other signaling molecules. And also if there's flow, like vessel gets stabilized, but like if there's no flow, some vessel cells just die. And in contrast to modeling blood flow in arteries, like this might be somewhat easier in one sense because flow is more laminar and there's little change in flow rate between a single heartbeat cycle. But the structure of a blood vessel is way more complicated than that of the artery. So this may require different approaches, and there are multiple approximations suggested in the literature. So like what we should do is to really figure out what are the common parts of those existing approximations. So what should be supported by the framework, and what should be determining what should be supported by what should be provided by the users. That's the part of our future work. And currently, updating single cell state is mostly up to users unless they want to do that by solving deterministic ODEs. This is bad in two regards because this asks users to do more coding, and especially writing solvers are difficult coding. And also, if users write more and more code, they will likely to write more and more slow code, and that will negatively affect performance. So we plan to provide solvers for popular approaches, and for very complex model, users may adopt different solvers and different combinations to model the whole cell behavior. So seamlessly supporting that kind of combination is our goal to improve Biocillion. And currently Biocillion maps a cell to a discrete agent, but to better model cell shape, one may need to map a cell to more than one discrete agent. for example, something like budding yeast, like it might be much better models using 2 cells instead of 1, and also other cells with non-trivial shape like dendritic cells, or F1 is studying epithelial to mesenchymal transition and like mesenchymal cell invading healthy tissue. Like mapping a cell to more than one particle may allow them to model physical interactions with higher fidelity. And also we can, if a cell is mapped to more than one discrete agents, we can consider each sub-agent as subcellular compartment, and we can assign separate regulatory network for different sub-agents, and also we can model molecule exchange between sub-agents to model like molecular transport within a cell. And also, we currently run this mostly on something like quad-core workstation to 1,000-core cluster. But like leadership class supercomputers have millions of cores, and we'll have machine with billions of cores in coming 10 years. So scaling up this framework and also tuning this for new accelerator architectures that can be a part of our future work if we can get enough users to justify this kind of time investment. Like this kind of performance tuning can significantly improve performance, but they can be also very time consuming and labor intensive. So currently, Biocillion is mostly for serious computational biologists who knows how to do coding, but there's clearly a necessity to make this easier to use. And this is mainly Simon's interest in improving the accessibility of this technology. And you can read through the slides. And so basically, if I conclude, I think for biological system modeling to be successful and to really answer questions we should answer, like the modeling framework should allow incremental updates and also validation by direct comparison with wet lab experiment in the same scale. And high-performance computers in combination with Biocillion can support that, but really to get there, there are many remaining works, so this requires further investment. And you should be able to find more details from biocillion.com, but currently that homepage doesn't have that many contents, but we're clearly gonna add more and also gonna release this software through the homepage sometime, hopefully pretty soon. And here's my acknowledgement slide. Like this has been supported by the lab, and like I think we are running out of time. But like if you have questions, I'm happy to answer those.
Thank you very much, Seunghwa. We'll open up to questions at this point. Any questions to the speakers? Please unmute your microphones and show your videos.
I think here's a question from Glenn. So the question is, do you currently or plan to allow users' models to be tracked, curated, iterated separately from the Biocillion framework? So like what's the question exactly?
So I think he's asking whether we expect to organize the models that people produce, like providing a library.
Providing a library. So that's something Simon is trying to do. So like through the Biocillion portal, like facilitate users to share their models. And if we can build a wizard, like users can start from existing model routines, then can modify that routine based on their need. Or like, so like that's something we are planning to do through like Biocillion importer or building a wizard.
I have a question.
Thank you. Yep.
Can you hear me?
Sure. Yep.
So essentially, your model only consider rigid cells, right? Not for deformable cells.
That's up to users. Like Biocillion framework does not really force anything about whether a cell is rigid or deformable. But like, it's not part of the framework, but it's a part of the model. So like, if we consider a cell as sphere, and like if we model shoving, if there's overlap, then we are considering a cell as something rigid. But like by applying different rules, we can also consider something more deformable. But like Biocillion framework does not provide or force any specific mathematics for that. So that's mostly up to users, not to the framework. So like if you can bring a proper mathematics, you can do that.
But it's not trivial to determine the cell-cell interaction and cell environment interaction.
Yeah, that's clearly not trivial. Yeah, so like bringing that mathematics is something highly capable and like computational biologists should do. So like if you really want to bring that mathematics, so like my key assumption is like determining those rules, computational biologists should be better than HPC experts. So that will be the role of computational biologists using Biocillion. But Biocillion's role is providing flexibility to integrate that kind of mathematics if they can come with that kind of mathematics.
I have a question. Can you hear me?
Okay. Yep.
Yep. This is Gary on from Chicago. So can you interface with SBML models or CellML models with this?
Currently, no, but that's something we are also considering to improve the usability of Biocillion. So currently, single cell modeling is mostly up to users, and users should put some code there. But maybe in the future, we can add SBML interface.
Well, like say we intend to add SBML interface in the next year or so. Really, that's
something we can do.
One of the projects we have lined up.
Currently, it's not possible.
Another question is, to my understanding, the agent-based model does not really realize some basic physical principles such as mass conservation, momentums, and dissipations. So I don't know whether you have any thought about that.
So like basically, models are really they are sort of good enough for their purpose, but like I really don't say they are really very mature. So for some more realistic modeling, smart computational biologists should bring a better model than what we currently have. But like but my key focus is to design the framework to support that kind of model if someone can bring such a model. Instead, and like I'm not really trying to say I have the best models. Models are mostly, for me, test cases for the Biocillion framework. And my hope is someone else can bring better models than something we already have.
But I guess something like momentum becomes just another state variable. I mean, that's the answer. Maybe to address that question.
So I'm really not claiming that the models we have are the best. And mostly models are even not mine, from our collaborators. And they may say it's best, but like, I really don't know those models to the enough level to claim whether that is something exceptionally good or something that isn't as good.
Have you considered GPU?
That would be excellent.
Have you considered GPU computing?
So, like GPU, you're talking about NVIDIA GPUs, right? Okay. No. So, main reason I'm not considering NVIDIA GPU is like here, we need to run both user routines and solvers. So NVIDIA GPUs may work well for solvers, but user routines, those are highly irregular, and GPUs are really not that good in executing that kind of code. So like I'm considering more of Xeon from Intel as accelerator coming from Intel, and like it can execute arbitrary x86 code. But the problem with the current version of Xeon nice corner is like the single-threaded performance for irregular application is pretty bad. But Intel is trying to improve that in their next generation architecture, Knife Landing. So I'm thinking that may work better for Biocellion than NVIDIA GPUs.
And for the multiscale time step issues that you mentioned, have you considered implicit scheme to kind of take care of the steepness problems in your systems?
So like PDE is implicit. We use an implicit method. PDE solver is implicit multi-step methods, and like if you can find more details if you go to the Chombo website. You're asking about this or asking about something else?
Yes, that's what I'm asking.
Yeah. It's an implicit method, so it can handle stiff problems. Okay, yep.
Hey, here again. So I noticed in the examples that it looked like cubic three-dimensional grids could use support other geometric topologies like dodecahedral or tetrahedral spaces, or does it not matter?
I cannot hear very well.
Okay, so I noticed that in the examples they were all cubic grids. So
okay.
would you support other geometric partitions like dodecahedrons or tetrahedrons kind of thing? Because some people like those spatial representations better.
Cubic grid. But like if you have different shapes, something like cylinder, it should be approximated as a combination of cubes, and we do not directly support cylindrical simulation domain.
Okay. Yeah, because there was the neighborhood artifact and the cubes.
Any other questions?
It looks like there is a question on the chat box where the visualizations you showed integrated with the Biocellion framework.
So like we are currently using ParaView, and we are also considering adding support for Visit. But currently, like this generates output that can be visualized using ParaView.
Okay, are there any other questions or comments for the speakers?
I have a comment. So you probably want to check out some groups. For instance, Professor George Kanialakis at Brown University. He used dispersive particle dynamics to model red blood cells. And another professor at UIUC, Jonathan Fruit, also used particle-based approach to model deformable cells.
Okay, thank you. Like adding those more complex models can clearly improve modeling, and thank you very much for your comments.
Great. Any other comments or questions for the speakers? If not, let's thank the speakers again
Thank
for
you.
that presentation.
Thank you very much.
Thank you.